Hello, everyone. Welcome to Foresight's Longevity Biotech Group, sponsored by 100 Plus Capital. And a few bits of housekeeping before we get started. One is we are having a AI for Bio longevity workshop in Berkeley on May 9th and 10th. So if you're interested, I'll put some information for that in the chat. And additionally, I think we have a couple new faces in the meeting today. So if you're interested in joining the biotech group, we meet monthly. You can email me at lydia@foresight.org. But today we're really lucky to have Dr. Amy Proal with us today. And she's Chief Scientific Officer of PolyBio Research Foundation, and she leads the LongCovid Research Consortium. And today she'll be talking about how chronic infections and the human virome impact health span and longevity. So if I missed anything, feel free to fill everyone in. But thank you so much for joining us, Amy.
No, that's great. And thanks for having me. I look forward to this. So I will talk today about the human virome as a driver of aging. And by the virome, I'm talking about all the viruses that persist in the human body that people acquire over the course of a lifetime. But I'm not even just going to talk about viruses. I'm going to also talk about a little bit, to the extent that I can, about the persistent bacteria and fungi and parasites that people accumulate over the course of a lifetime, and how all of these persistent pathogens can contribute to many different forms of aging processes. So here on this slide, I grabbed this from a talk that I saw someone present recently on aging. Hallmarks of aging. I'm sure everyone is familiar with all of the ones on this list: senescence, mitochondrial dysfunction, chronic inflammation. Now, what a point I would like to make, and that is the point of my hall talk, is that persistent pathogens, viruses, bacteria, fungi, parasites can cause every single thing on this list to happen. They can literally, and there's dozens of examples that I could give you for each of these bullet points, just proof of concept of a pathogen that, yes, shortens telomeres, yes, causes mitochondrial. So that's what I'm going to walk through. And really then what we need to do is start to think a little bit more about how all these pathogens that we accumulate in our bodies are contributing to all these really hallmark of aging processes. So these are the ones I'm most going to focus on in the talk: inflammaging, mitochondrial dysfunction, senescence, telomere attrition, epigenetic alterations, and just cognitive decline. All right, so the human virome. Again, what I mean by that is that starting in the womb, where we often acquire viruses actually passed to us from our mothers in the womb, and other pathogens often, or even via the father, sperm can harbor organisms that actually seed an infant during development in the womb. So we are already often not sterile, and pathogens are part of who we are as an infant. Then we're born, and over the course of a lifetime, as everyone knows, we acquire more viruses from just close contact with other people, from the foods we eat, from water, from all kinds of other exposures, from insect bites, from many different vectors. And as these viruses accumulate in us, many of them don't clear. So key is to understand that, for example, all of the herpes viruses, and a prime herpes virus, for example, is Epstein-Barr virus. I think most of us know Epstein-Barr virus. Once you get it, right? Maybe you're in college, you get mono, it's a thing for a while. A lot of people then forget it. They're like, my mono's gone. Actually, though, you have Epstein-Barr virus in your body for the rest of your life. It does not clear. It is a persistent virus. So most of these viruses, the herpes viruses there's other viruses, the adenoviruses, the anelloviruses, the papillomaviruses, most of them which can persist with us for long periods of time. Now, the extent of which of this virome is growing too. This is a study that I love from a team in Japan. And what they did is they got tissue from people who unfortunately were passing away due to accidents, basically car crashes or sudden death events that didn't have something or anything to do with chronic disease. So they're considered healthy individuals that passed away. And they got all kinds, I think it was 39 different tissue types from these people. You can see that on the top of the graph. So we have all kinds of tissue: thyroid, breast, heart, liver, vaginal, testes, skin, muscle, thyroid. And then on the left, viruses. And these are the viruses, you can see in the middle boxes, that were found in the tissues of these healthy individuals. And at the top of the list, you see the herpes viruses, which is expected: EBV, HHV6, HHV7. Then, though, you even start to get into RNA viruses that most people just don't think persist and don't really consider are persisting in humans. For example, a decent number of tissues have RSV in them. Another set of tissues have a coronavirus, HCO229E, which is a coronavirus that still circulates, I guess, before SARS-CoV-2. And then other viruses that we see at the bottom that are really less studied. And the techniques that the sequencing this team used are really good. So these are unlikely to be contaminants. These really were in people's tissues. So the scope of the virome and the viruses people can harbor is pretty vast, especially when you consider that these people were healthy. That's just viruses. And Korean, I see your hand.
Yeah, in that previous chart there at the bottom were these things like tomato viruses and deformed wing viruses. Those aren't even considered human viruses, are they? But here they are in humans.
This is a question about this study. People have gone over the sequencing. They use metatranscriptomics, where they pull out all the human reads from the sample, and then they map whatever is left back to basically whatever is remaining that isn't human. They map to databases. I agree with you that some of the ones at the bottom, like tomato spotted wilt virus, seem unusual to be in humans, but people don't know anymore. There's a point where people wonder if it's not impossible that sometimes via some sort of food exposure or some other thing, it's possible that someone could harbor one. So I think that the ones at the bottom require further exploration. No. But you're right, they're a little weird. Yeah. For sure. So anyway, let's see. Okay. Also, those are viruses, but really parasites, for example, are also chronically acquired by many people. And toxoplasma just being one of the most obvious examples. 30 to 50% of the global population, depending where you live, harbors latent toxoplasma or supposedly latent toxoplasma in the central nervous system often. So we're talking about in the brain. And of course, this is a parasite that can drive all kinds of neuropsych and neurocognitive symptoms if it's active. So this is just another example of a type of pathogen that people carry that we don't sometimes think as much about as I think that we should. Next, we have just on this list here, I just wanted to add in some bacteria, tuberculosis, mycoplasma, bacterial organisms that many people harbor persistently. Fusobacteria have recently been found in endometriosis tissue, for example. P. gingivalis has been found in the Alzheimer's brain. It's a big driver of tooth decay. Chlamydia pneumonia has been found in atherosclerotic plaque. Staphylococcus is another pathogen that persists. And there's just a lot of bacterial passengers that people can pick up. I wanted to just add some of those in there just to make sure that people understand it's not just viruses. Okay. Then, this is just an interesting study that was actually done by Stephen Quake's team at Stanford several years ago, 5 years ago. And they used cell-free sequencing of cell-free DNA, which is a cool method to pick up reads of different pathogens. And this with blood from healthy people. And this is another study that sits there that needs follow-up. When they were using this cell-free sequencing, they found really thousands of new sequences for different pathogens, bacteria and viruses in the blood of healthy individuals. And so this is a little bit of the world in which, look, we have our kind of known pathogens like the herpes viruses that are obvious members of the virome in these communities that stay with us. And then there are teams still exploring other organisms, some of them surely pathogens in the human body that has still yet to be characterized. So there is an ongoing, and that's another thing to emphasize, just exploration of just the sheer number of organisms and pathogens that can be in humans. So overall, just do this here. Now, I'm going to go into just mechanism here. One of the obvious drivers of aging is inflammaging. You have more inflammation, unproductive inflammation over the course of time as people age. So obviously that's an issue. Now, one of the things, for example, that you'll see in this study is that IL-11 is a cytokine. It's an inflammatory signaling molecule. It can promote inflammaging, right? So in this Nature paper in mammals, I believe it was mice, they inhibited IL-11. It's actually a pretty straightforward study. And that extended the lifespan or the health span and the lifespan of the mammals in the study, which, is a pretty basic experiment, but shows that you're mitigating this inflammaging that does have an impact on health span. Okay. The big question then is what activates cytokines? What is kicking them up? What's their activity? And the number one, driver of cytokine activation would be infection. Every single pathogen, every single one, if it does anything in you, if it creates any protein, any metabolite, if it works to just replicate, get into a new tissue, it will activate cytokines in the process because that's how the immune system is recognizing the pathogen. It's creating cytokines to try to keep it in check, to mitigate it. So if you see cytokine activation, that's a strong sign that infection could be involved. So for example, here is just a proof of concept study showing that respiratory viruses kick up IL-11, which is the cytokine we saw in that study that if you inhibit can be associated with extending health span. So obviously, like, pathogens as drivers of inflammaging is a no-brainer. Then we get to the mitochondrial dysfunction. And this is a paper I wrote with my colleague at PolyBio, the nonprofit that I run, Mike Van Elziger. We wrote it a few years ago. What we wanted to show in the paper were concrete examples of how essentially every pathogen, viral, bacterial, fungal, hijacks the mitochondria of the cells that it infects in order to replicate in the case of viruses and to gain substrates to create essentially new backbones. And in the case of bacteria, to replicate, but also to gain nutritional substrates for them to be able to live on better. This hijacking of host mitochondria by pathogens in order to gain the substrates and other nutritional metabolites or things that they need for their own benefits is profound, especially in the case of viruses, because viruses are obligate intracellular parasites. And what that means is that they cannot create another version of themselves without pulling substrates from our human mitochondria. They simply have to. So every single time that a virus replicates, including a persistent virus that might be persisting in someone across the course of decades, it will by definition alter the mitochondrial output of the cells in which it is persisting in. And that would inevitably lead to altered mitochondrial output in someone who has those issues happening. So obviously there is a huge connection between mitochondrial dysfunction and persistent infection. Okay, then here, this is just for an example. This is a figure from the paper. The blue boxes have different viruses in them. Those are abbreviations for different viruses. And these are just different parts of the mitochondrial pathways that are intending to create ATP for our own efficient energy use that are hijacked by pathogens. Often what the pathogens do is push the mitochondria into basically a Warburg metabolism, where it's less efficient, they're producing less ATP, and it's more inefficient, more lactate is produced as a result of output. So it's an inefficient type of metabolism that they push towards. So that's another thing that pathogens do that can impact aging processes. Then just going into senescence, many pathogens create
Yeah, thanks. The previous slide there, is it not also the case that in the glycolysis Warburg state, the metabolic cycles produce more amino acids and nucleic acids and all these kinds of things, and therefore the viruses like that because they need amino acids and nucleic acids to build themselves, whereas in a oxphos state, you're getting more ATP and less of those other things. You're doing more energy production and less proteosynthesis and stuff like that.
That's exactly correct. They all are pushing the mitochondria towards making more byproduct that they can use. A good example, and I'm going to go into a couple more of those examples later, is more lipid synthesis, for example. Viruses love lipid, and so you'll see mitochondrial metabolism pushed by a lot of these pathogens into the creation of more lipid droplets that they then use for parts of their replication, for parts of, they hide in the lipid sometimes. They create rafts out of the lipid to spread from cell to cell. So a lot of what's happening is exactly so that they can have more of these products for the things that they need to do.
So I don't mean to divert us, but I just want to ask one more question, if you don't mind. But that is curious to me because from what I understand, we also have these autophagy processes that go on to create, not create, but to recycle some of these substrate molecules like nucleic acids, lipids, aminos, and stuff like that. Is it that the viruses cannot leverage those?
I'm not sure. You mean that like our own cells are all, like, explain again.
Our own cells do not get all the nucleic acids, amino acids, and lipids that they need outside of the ones you get from your diet, the essential ones. They do not get all the other synthesized ones by synthesizing them de novo all the time. They get a lot of them by recycling damaged molecules and taking them apart. And it seems like the viruses somehow can't leverage that and have to get de novo synthesized ones from glycolysis.
It seems like it, but to be fair, we only explored that direction in this paper. I'm not joking. I bet if some labs started working on how viruses or other pathogens can also take advantage of parts of autophagy and where there would probably be some way they exploited it. I don't know. It might not be impossible, but from the papers and the evidence we were looking at, the examples that we have so far are where they're actually just using the substrates more straightforwardly this way. Yeah. But it's not, I'm not sure. Yeah. It could be Creon that even they take advantage of extra effects. But yeah. So yeah, going into senescence, again, you can just Google like pathogens, senescence. A lot of the toxins, the products, the byproducts, for example, the bacterial cell wall components of bacteria, like liposaccharide, which is a component that forms the cell wall of, again, yeah, most bacteria, all are able to trigger inflammation in a way that can, in many cases, result in senescence. So this is just one example where a team showed that salmonella and a toxin produced by salmonella triggered a pro-inflammatory senescence-associated secretory phenotype, which is key under phenotype in senescence. You can have this pattern in which a pathogen will push a cell or group of cells towards senescence. And what I think is also interesting is there are other studies that show that senescent cells are often more easily infected. So there are groups that have done lab experiments which show that if they infect senescent cells in a certain tissue type and non-senescent cells, the cells that are senescent are more likely to get infected, stay infected, become infected more easily. So it's a feed-forward mechanism where the pathogen can push the cells into a state of senescence. Now those cells seem easier to infect, and you can see how that would be detrimental over time for the health of the turnover of the cell. So that's another one. Then here, I think we get into a paper that to me starts to really capture the heart of really what viruses and other pathogens are doing to most drive age distortion. Because driving inflammation is one thing. The hijack of mitochondria is a big deal. But really key of the way that pathogens can most mess with us is that they directly hack us. And what they'll do to do that, and that's what this study looked into, is create proteins. They'll infect us. They can get into the center of our cells. Many of them are intracellular. And then they create proteins and metabolites that can directly alter our own human gene expression. And that's what this study tried to capture. And I recommend reading it, this whole paper. What they did is they wanted to see if viruses could act as age distorters, to see if their reproduction benefited from interference with host aging processes. So they took a human longevity database where the database included all kinds of proteins that were basically receptors and other proteins associated with important human longevity signaling. So receptors that control apoptosis signaling, that control senescence signaling. Then they took a database of viral proteins and they just mapped together how many of the viral proteins could directly bind and distort and interact with the signaling from those human longevity protein networks. And there were just dozens of interactions. So this is just the two of the viruses, and you wouldn't be surprised with the first one. HIV is just a huge age-distorting virus. Interestingly, a virus that had a lot of age-distorting effects was H1N1, which was influenza. But if you look at these maps, the white lines are basically the white edges are the human proteins associated with the longevity networks. And then the blue edges that are going over those white lines are the the human and viral protein interactions that can directly affect those longevity networks. And then the different sort of orange and yellow ones just show, oh, that protein is associated with apoptosis. Oh, that one is associated with senescence. So there's this huge amount of basically interference where viral proteins can interact with human proteins in a way that can distort the signaling of those human proteins and their ability to control key longevity signaling pathways. Now, these are some of the top viruses that had these interactions. At the top of the list with 45 interactions that were the top for driving cellular senescence-associated phenomenon was the Epstein-Barr virus. So again, this is the thing, is like when you get mono and you don't think it's a big deal, the problem is that later on, that virus might actually be creating proteins and products that are really able to distort longevity signaling associated with senescence and other factors. So the team who did this study actually concluded off this, owing to the considerable number of human viruses. Oh, sorry, let me go back. Oops. Oops. One moment. Let me just. Owing to the considerable number of human viruses, this evolutionary-minded view encourages a reconceptualization of the locus of aging, no longer exclusively focused on our own genetic material, but expanded towards a larger set of genetic entities interacting with our species, such as viruses. I totally agree with them. What I want to do, though, is explain a little bit more of, again, like how it is that protein interactions can be detrimental to longevity. And what I mean by that is a viral human protein interaction. A lot of that comes down to molecular mimicry, which is a way of saying that many human proteins are very similar in size and shape to the proteins created by pathogens. So there's only so many structures that a protein can have. And so you can have a human protein and then an Epstein-Barr virus protein that has a very similar size and shape. And what it means is that often there's the human protein that's supposed to bind into a receptor that, let's say, controls senescence-associated signaling. What can often happen is that the pathogen protein that has a very similar size and shape to the human protein can also bind into that human receptor, except when it does, its fit is a little bit different, and it will actually distort the signaling of the receptor in a way that makes the downstream signaling wonky and often favors what the pathogen wants instead of the normal human signaling that would usually happen via that pathway. So a really interesting example of that is this team, which found that viral insulin-like peptides can activate human insulin and human IGF-1 receptor signaling. So what they did is they isolated this family of viruses called, I think it was Inflammaging. I have to remember the virus name. And they had isolated the viruses from human blood samples and I think even human fecal samples. So they are viruses that are capable of persisting in the human body. And what they showed is that these viruses make proteins that are really similar in size and shape to human insulin. And so then they did a series of really good experiments showing in mice and in models that those viral-like insulin proteins or peptides could bind into the mouse insulin receptors in a way that altered the downstream glucose metabolism of the mice, but in a way that fairly distorted it. In other words, the viral-like peptide could alter the murine or the host receptor signaling in a way that altered the glucose signaling of the organism. So this can happen in so many different ways, where a pathogen protein, again, is similar in size and shape to a human one and can distort almost any kind of human signaling network in the human body. And in this sense, this is what makes persistent pathogens and the proteins and the metabolites they create the closest thing to really being able to hack us. They can take control over our own signaling pathways and modulate them towards basically pathways or signaling that often benefit their own fitness over ours. So here then now switching to telomere attrition, here's just an example. For example, there are viruses that have been associated with telomere shortening. A good example is human herpes virus 6, which again is a persistent virus that stays with people throughout life. In this particular case, this team showed that HHV6 integrates into telomeres near the end of telomeres, actually. And in this particular study, the telomeres in which the herpes virus had integrated in were the shortest and the most unstable of the telomeres, suggesting that the integration probably was associated with telomere attrition. And one of the things that's interesting is that the integration of the herpes virus 6 into the telomeres is actually part of how the virus maintains its latency or its ability to remain fairly dormant in the body. In other words, it's actually using integration in the telomere as a mechanism by which to stay dormant and somewhat hide out from the host immune system. And a number of herpes viruses share that common mechanism of having herpes virus integrated telomere proteins as part of their latency. So there is obviously a relationship between our telomeres and their activity. Now, getting to the epigenome, could go a whole talk on this one. There's many different mechanisms by which pathogens affect methylation patterns, chromatin remodeling, all kinds of things, again, via the proteins and peptides that they create. But for example, as just a very simple study, you can see papers like this exist where, for example, this team did find that people who harbor cytomegalovirus simply had accelerated epigenetic aging as assessed by just epigenetic markers in the study as compared to people who didn't harbor the virus. The same has been noted for people who have gotten COVID several times. They seem to have accelerated epigenetic aging compared to people who didn't get as exposed to the virus over time. So there are pretty clear connections between people who harbor persistent viruses and certain changes in accelerated epigenetic age. Okay, so then I wanted to also get into the cognitive decline aspects of what viruses and other pathogens can do. This is a study that I think is interesting. It was a team who looked through hospital records of a really large data set of people. And they found that viral and actually to a point bacterial and fungal infections, so they went back 10 years before at least, and they started to track who had more infections via the databases and compared. And people who had more infections, bacterial, fungal, or viral, were more likely to develop Alzheimer's disease and Parkinson's disease. Those were the two strongest diseases where there was a pretty strong association between increased number of infections and the development of those chronic conditions. This is a similar paper that did the same thing. It was a team that again mined medical records and found that people who had been exposed to these pathogens and had sustained these infections more often were, again, more likely to have not just Alzheimer's disease, but there was some signal with ALS, dementia, multiple sclerosis, which is increasingly connected to viral bacterial activity via a number of studies, and even parts of Parkinson's disease as well. But when you get more into, and at this point, I wanted to pause and say, you're seeing here respiratory viruses like influenza. And I feel like that probably is confusing to people because you would say, okay, I guess I understand how I could get influenza. And during the time that I have the flu, some of these changes that the virus could do that happens during acute infection could affect my brain, could affect my mitochondria, things like that. But one of the trends that we study a lot at our nonprofit and that we've actually been studying a lot in people after COVID is the persistence of respiratory viruses as other viruses that are capable, at least in some people, of not fully clearing from their tissues. So for example, this is a group of us that have been working on LongCovid, so people who have chronic symptoms after COVID. But the key thing we've been working on in these patients is understanding if they just don't clear the SARS-CoV-2 virus over time. So this is a paper we published about a year ago. And what we were looking at is, again, this very basic explanation for why at least a certain number of people continue to have chronic symptoms after COVID. And that's because we find in a growing number of studies that they still have little bits of the SARS-CoV-2 virus in, let's say, the tissue of their gut or in their lymph nodes or in other sites that we basically call reservoir sites. Not as often in their blood, but in little sort of tissue-associated reservoirs where the virus is better protected from the immune response. So in other words, SARS-CoV-2 is capable of acting as a persistent virus, at least in some people. So for example, this is a study published fairly recently in Science Translational Medicine, and this by a team at University of California, San Francisco. And this is an image, this is gut tissue taken from the colon tissue, basically. This is a person who had symptoms after COVID. But you can see here, for example, in the image all the way on the right, this is a person 676 days after the only COVID infection that they knew they had reported having sustained. And the pink there is the backbone of the SARS-CoV-2 virus. In fact, that's double-stranded RNA, the pink. Double-stranded RNA is RNA that's only produced when the virus replicates. So if you find it in a sample here, it does strongly suggest that the virus was active and was replicating in the tissue at the time at which it was collected. And then the other sort of purple, lighter purple blobs around the pink are immune cells that have come in and infiltrated their way around the persisting double-stranded RNA of the virus that's still in the gut tissue, to the point where the immune cells are clumping in around the RNA in a way that's almost like a granuloma. So this group actually called this a coronaloma, which is actually disturbing. We don't know how many coronalomas there are in people's guts, but it actually is a thing now where there's a lot of data increasing showing that the SARS-CoV-2 virus especially doesn't clear from the tissue of the gut because the gut is so dense with ACE2 receptors that the virus uses for entry, and the gut is fairly tolerant to organisms, so it doesn't have sometimes as strong an immune response as other body sites to really eradicate every pathogen that gets into that site. you can have then persistence of respiratory viruses. This is just one more interesting example, I think, with the persistence of SARS-CoV-2. This is a study done by a group who got tongue tissue. They did little biopsies to get pieces of people's tongues who had loss of taste after COVID. And in every single person in the study who had loss of taste, they still found the SARS-CoV-2 virus in the piece of the tongue tissue. So there was clear viral persistence in the tongue tissue associated with inflammation. So again, like loss of taste could actually be connected to viral persistence. And this is just from that paper I showed you before. These are just more studies that have found that SARS-CoV-2 can persist in tissue or in some cases blood, for months or years after initial infection. The interesting and concerning part is that not everyone in these studies has symptoms. And in a lot of cases, we do find persistent RNA or protein from the SARS-CoV-2 virus in people without symptoms, and we don't know what that means. It could mean that it's not a big deal, or it could mean that, like other viruses, it might remain dormant for a while but cause disease at a later date. So that's something that we continue to study. And this is a statistic or a paper. This was in 2022, but it was reported by a team that Alzheimer's disease risk did increase by 50 to 80% in older adults who caught COVID. That's a pretty disturbing statistic suggesting that SARS-CoV-2 could be contributing to increased Alzheimer's rates. And I'll just go into a little of why that could be now in my next slides. Because what I'm showing you here is actually, I think, some of the most important research that has occurred in this basically world of neurocognitive decline and infection. And this is work from Rudy Tanzi's lab, which is at Harvard Medical School. And this is a paper where they showed that the plaque that accumulates in the Alzheimer's brain, the amyloid plaque that we blame for the disease, actually in their models, and they have neuron in a dish models, they also have mouse models, what they've shown is that it seems to have a function. It actually functions as part of the innate immune response that forms in response to bacterial, viral, or fungal pathogens that get into brain tissue. In other words, if a virus, and in this case, what they did was they put herpesviruses into their neuron in a dish model. If the virus is put in the dish, the amyloid plaque forms as part of the immune response to try to contain that virus. And it almost ingests the virus in a form of hydrogen peroxide or bleach so that it's now trapped inside the amyloid plaque. So what these papers show is a potential paradigm shift in what causes Alzheimer's disease, which is the plaque might form for a reason. It might form in order to contain infection. Thus, it has a purpose, right? So this is a study done by the same team, which again, in their model showed that the amyloid beta plaque formed in response to infection with microbial infection, so with bacterial infection, and even in a fungal infection model. So basically, it seems like any pathogen can see this basic innate immune response that could be forming Alzheimer's plaque. So one of the key things there to consider is that it does make sense as a model that amyloid plaque would have a function like this. Because if you look at amyloid plaque, even zebrafish, which are really ancient organisms, can form amyloid plaque. And you would think then that if plaque was only detrimental, that if amyloid plaque simply accumulated as just a pathological process alone, that it actually would be weeded out over time so that humans wouldn't retain the ability to make amyloid over such a long course of sort of evolution over time. But humans do form amyloid, again, suggesting that this conserved ability to form amyloid plaque is likely because the plaque has some kind of function, again, which seems to be tied to its ability to contain infection. So there's a lot of teams that are following up on this now. This is a really interesting study that came out very recently. This is Ben Reedhead's work at Arizona State University. And what he did was he got tissue from the gut wall of patients with Alzheimer's disease, and this was tissue from people who had passed away from Alzheimer's disease. Then he got tissue from the same people's vagus nerve, which is a nerve that connects most of the major trunk organs of the body to the brain. And then he got also tissue from the brain. And what he found was in Alzheimer's patients who had a specific CD8 like 83 positive microglial subtype, which was a certain kind of pro-inflammatory activated microglia, those patients specifically often had cytomegalovirus, which is a persistent herpes virus, in the tissue of their gut, in the tissue of their vagus nerve, and then in the tissue of their brain localized with Alzheimer's plaque. And what they concluded from the study is that it's very likely then that the virus may begin in the gut, right? That it might start by just infecting the gut of someone and be in that area of the body for a while, but that over time, it may literally crawl up the vagus nerve, using the vagus nerve as almost a highway into the brain. And once it gets into the brain, then it might be able to seed Alzheimer's plaque. And by the way, these are images of cytomegalovirus there. That's in the sort of green-yellow, concentrated within the microglia around the plaque in the Alzheimer's brain. And these are images of cytomegalovirus on the top slides here. And you can see especially the profound imaging of the virus on the left and the slide to the right of the upper slides in the vagus nerve itself, which is really incredible. So what I wanted to then mention is going back to that mitochondrial hijacking that Kriyan had actually brought up before, which is that when pathogens hijack mitochondria, let's say when they infect a brain cell and they're going to inevitably affect the mitochondrial output then of that cell, they often create more lipid as part of that hijacking process. Now, one of the things that I think is interesting is this study fascinated me. It's a team that found lipid droplet accumulating microglia were extremely common are extremely common in the Alzheimer's brain. So up to 50% of microglia in the Alzheimer's brains that this team studied, I think they're at Stanford, contain these lipid droplets, right? And these lipid droplets were extremely pro-inflammatory, and the lipid droplet cells were expressing more reactive oxygen species and were more basically pro-inflammatory than some of the other microglia in the brains. Now, again, I just want to emphasize that lipid droplet hijacking by pathogens is just a common mechanism. And so anytime that you see a lipid droplet, you get to the point where you want to say, is that cell infected? Because, for example, here's just another example. It's an image of the parasite Trypanosoma cruzi, here in the green, inside again, or near the lipid droplets of a cell, right? So one of the things that we're trying to encourage that group with the lipid droplet microglia in the Alzheimer's brain to do is just sequence the lipid of those microglia and see if it contains any pathogens in it. And I suspect that it does. So now we go into this sort of, what do we do about it? If these pathogens can do this, if they can persist in us and drive inflammaging, senescence, epigenetic alterations, if they can hijack our signaling pathways, if they can cause all these issues, what can we do to control them? And there's a lot of really actually very basic things that we can do. There's so many things that we can do actually that I could not even begin to do a whole talk on this. But this is an interesting study that just shows that even existing affordable antivirals could go a long way in preventing dementia. It's a group from Taiwan. And again, they went and they got a population level database of medical records from people. And what they did is they looked back at people who'd been given basically Valtrex or other herpes virus antivirals that are prescribed commonly for cold sores or sometimes for genital herpes. And they looked at the rates of dementia in people who had taken more of those basic herpes virus antivirals versus people who had not. And the people who had taken more of the herpes simplex antivirals had a significantly lower rate of dementia, which strongly suggests that actually just existing basic antivirals could prevent dementia risk. So actually what we're doing is Ben Reedhead, who's the researcher who did that work on cytomegalovirus in the vagus nerve and the brain, we're working to get him support to run really one of the most low-hanging fruit clinical trials, I think, ever to exist, which is to run a clinical trial of the existing antiviral valacyclovir in patients with pre-dementia to prevent progression to Alzheimer's disease. And it's a great trial because patients will measure actually the p-tau biomarker. I think some of you might know about that. It's a blood-based biomarker that can track tau now via blood test, which is cool. and this other biomarker he's been able to develop that can capture response to cytomegalovirus fairly accurately, again, from blood. So in the trial, he'll actually be able to track not just if people's symptoms improve when they're taking the valacyclovir antiviral, but actually if literally these markers improve. He's even doing some forms of FDG PET imaging and other things so that you can really say at the end of the trial, yes, this person does not have dementia in the way that they had before. And some of the pilot data that he has does show actual reversal of these markers from pre-dementia already in certain people who have undergone this antiviral treatment. Then, what I think is interesting too is, I guess a lot of what I wanted to mention is that a lot of the supplements and medications being used already by the longevity community to promote health span are some of the same medicines that we use in the world of controlling infection-associated chronic disease to control infection. And so to make that point, what I put on this slide is I pulled some of the top medications and supplements that you can get through Ageless, right? And those are, let's say, low-dose rapamycin, low-dose naltrexone, antifibrinolytic enzymes, glutathione is an antioxidant, methylene blue, which a lot of people are taking, NAD+, methylation-based support. All of these things that people are doing for longevity are actually, interestingly, the top treatments that we use at our clinic. And part of my time, for 1/4 of my time, I'm the scientific director of Mount Sinai's, basically it's called the Core. It's a clinic. We've existed now for about a year. We're a new clinic innovating care for patients with chronic disease tied to infection. And the top treatments we use are low-dose rapamycin, low-dose naltrexone, antifibrinolytic enzymes. We use basically the same treatments that a longevity community uses. In our cases, though, we're using these meds because of data showing that they can control infection. So if you look at rapamycin, this is a really interesting study. This is basically, I think, a group that used a rapalog, RAD, RAD001, an mTOR inhibitor. And they found a couple of things that were interesting. They gave the people it was elderly people who were healthy otherwise, who took the rapalog for, I don't know, a couple months, I think. At the end of taking the rapalog in a low dose, they gave them the influenza vaccine, and they had a more robust response to the influenza vaccine, suggesting a more robust immune system. Then what I thought was really fascinating from this paper is that they measured interferon gene expression after people had completed the time of taking the low-dose rapalog. And they found all these different interferons genes were upregulated. And now interferons are basically maybe the most important components of the immune response that keep viruses in check. So the interferons, for example, keep herpes viruses in latency. So if you can upregulate interferon signaling and keep interferons in a good state, you can essentially keep herpes viruses in a more dormant state. So strongly suggests that there could be an impact on low-dose rapamycin of actually keeping interferon signaling in check and thus pathogens in check. This was another study, similar study by the same team. What was interesting in this one was that after taking the rapalog, there was some decrease in T cells that expressed PD-1, which is a marker of T cell exhaustion. So the T cells seemed to have a more robust, energized phenotype, which is interesting. Again, that's what we would see if we wanted to better contain infection. We want our T cells to be not exhausted. We want them to be active. So the fact that perhaps this rapalog improved T cell exhaustion is actually pretty amazing. And then just going into these studies, we use methylene blue in the treatment of people who actually have chronic tick-borne, vector-borne infection. For example, when people get Lyme disease or co-infections like Bartonella or Babesia, which ticks can pass to them, the main treatment that we use as an antimicrobial is methylene blue. It also has antiviral activity. It's being used in COVID. More people are actually starting to take methylene blue. Yes, when they get COVID, it's been used in the treatment of dengue. It's been used in the treatment of Zika virus. So methylene blue absolutely has antiviral, antimicrobial activity. Also, metformin is one of the bigger drugs that we're using in the LongCovid world. There's actually the study that most shows treatment during acute COVID to prevent people from developing long COVID is a trial in which patients were given metformin. And the patients who took metformin when they got COVID were much less likely to get long COVID. So there is an effect of metformin on controlling viral activity. The mechanisms are not perfectly sorted, but honestly, these drugs like metformin or even rapamycin that are affecting mTOR function and thus having, or in the case of metformin, are also having other effects on mitochondrial metabolism, it could be that parts of the way that the drugs act get in the way of how pathogens can hijack the mitochondria. That could be at least one of the ways in which some of these drugs are able to have kind of an antiviral, antimicrobial, or anti-parasite effect. So basically, that's where I was going to go through. I don't think that we will succeed in expanding lifespan and health span until we start to stop this hacking of chronic pathogens. I think that right now, if someone had a computer and it had a virus, we would understand that we could replace the hardware, we could put in a new hard drive, we could make the laptop a lot better, we could do all kinds of things to it. But if it has a virus that's still messing with the code of the laptop, there's just no way this is good. And so I think that one of the key things we need to do is to both create better diagnostics to be able to understand what persistent pathogens people have in them and what they're doing to our metabolism and to things like senescence and apoptosis, and to be able to incorporate those diagnostics into some of the age-associated tracking that people are doing over time. If we begin to do that, I think we can normalize the fact that we can begin including infections and the chronic infections especially as just some of the factors that are contributing to changes in health span. And then we can begin to bring in, honestly, a cornucopia of antivirals, whether it's drugs or herbs or other treatments or all kinds of different therapies that people can use in a preventative fashion to actually keep the organisms or pathogens in them from being able to do a lot of the hacking that they would otherwise do if we didn't work to keep them in check. So that's my message.
Thank you so much. That was so fascinating. And I also feel like I have a little bit more to worry about now. But I have so many questions, but I think we have a lot in the chat. Josh, would you like to ask your question? I can also read it from the chat. Okay. Josh asks, it's in the virus's interest that we live for a long time, so we can continue to spread the virus. Are there any cases where you've seen viruses activate longevity pathways?
No, I'm not sure. I understand the thinking that they would want to just have a host that just lives on forever. At the same time, different viruses have different strategies, right? Some kill you. Ebola just wants you dead, right? Other viruses want to persist in you and see if maybe they can create a longer term atmosphere. So first of all, it depends on the virus. Not all of them necessarily always move towards extending the time or whatever fitness of the host. I don't know. This is the interesting part. I don't know of any examples in which a pathogen seems to have beneficially affected someone in a way that could contribute to longevity. The truth is that I'm totally open to finding some examples of this, but at the moment, most of the examples that exist in the literature go the opposite direction, where the pathogen is doing something more detrimental than it is helpful.
Can I follow up?
Yeah.
I think we need to distinguish between natural viruses, which were evolved for their own survival and reproduction, and bioweapons.
Yeah.
Ebola is a bioweapon. It came from a lab. SARS-CoV-2 is a bioweapon, and they are not necessarily created to promote their own longevity. They are created for the express purpose of doing damage to the body. When other viruses, natural viruses, do damage to the body, it's incidental in the process of reproducing. But when a bioweapon creates damage to the body, that's because that's what it was engineered to do.
That's true. And I think if you look also at some of the herpes viruses and some of the other pathogens that are players in aging, The truth is that there are some examples of which they've been, for example, like in injectable medicines, or there's an MIT paper that I can send out later to the group that does show that every now and then a pathogen does get out into the population, again, via just, it didn't come from an animal. It came from a lab experiment. It came from passaging. And there's some of the herpes viruses even come from that angle as well. So there is enough of a consideration. And I think that's one of the things where our team has been studying LongCovid, and it's hard now because everyone's burned out on COVID and we get this so much, right? But when we see the SARS-CoV-2 virus still in someone's tissue, part of that is because it's a wonky virus and because it was designed to evade the immune response. And it is, you know what I'm saying? That's part of why the persistence of the virus is likely happening, is because there is a wonkiness to it. And I think that what we hope is that in the future now, as like potentially the NIH and other groups think about what they want to fund in terms of understanding what chronic viruses do and chronic disease, that maybe we're able to say some of these things out loud a little bit more in terms of the way that we're able to approach studying them, because it does inform what they can do and some of the mechanisms of what they can drive.
Thank you so much for acknowledging this. There's a lot of head in the sand refusal to acknowledge the bioweapons. As you know, Lyme disease is another prominent bioweapon that's endemic in the population.
And again, I don't know to what degree they were like deliberately created. It's more that just they were worked on in some way, right? And it could be an accident that they got out or whatever, but there was some form of laboratory-based manipulation to try to move them towards becoming more pathogenic. And often that involves making them more persistent. That's what's very worrisome for this, including in the case of Lyme. Yes.
I know we're reaching the end of our time block. Would you have time for a couple more questions, Amy?
Yeah, I can stand for a
Amazing.
more other people. Yes.
Thank you. I think next we have Nita.
Thanks so much for the talk. Just going off what you were saying about SARS-CoV-2 specifically, are there certain types of proteins that you feel are more responsible for driving some of these long-term effects? Like, I know there's been a lot of focus on the structural proteins like spike, envelope, membrane, nucleocapsid, but have you seen anything with like non-structural proteins or accessory proteins potentially causing some of these long-term effects? And is that something that you see across other post-acute infection syndromes?
Yeah, it's a really good question. The truth is the spike seems the worst, but people don't study a lot of the other structural proteins as much as spike. So it's a self-fulfilling thing because people have studied spike so much more than the other proteins that we just have more data on what the spike can do that's damaging. But the truth is that there's a couple things that make spike pretty bad, right? One, honestly, is the way it binds fibrin and fibrinogen. It just directly, there's 2 good papers out on this now. It literally latches onto fibrinogen. And that is difficult, right? Because then that starts to accumulate, that starts to cause clotting processes. That's going to cause all kinds of problems that increase the vasculature associated effects of the virus, which are some of the worst, right? So that fibrinogen sort of relationship with spike right now is one we've been concerned about for a long time. Also, spike seems capable of acting as a super antigen where it can sometimes really skew T cell signaling. So spike has a lot of its problems. So I'm going to go with the spike being the worst, but the truth is I think we need more study of the accessory proteins too. We don't want to count any of them out.
Okay, thanks so much.
Awesome. Noreen?
Yes, thank you so much for a really fantastic presentation, Amy. I was just blown away by your presentation. And I also just wanted to know, so what about beneficial gut bacteria or other microorganisms in the gut that have co-evolved with humans? Do you think that they could sometimes be pro-longevity because it might be in their interest to be pro-longevity?
Yeah, it's a great question. And I didn't have time to put the microbiome into this talk. Obviously, what I was focusing on are just the single pathogens, right? Like your single viruses and the single bacterial pathogens, the kind of like bigger drivers. But obviously we have these trillions of other organisms in and on us. Now, I think one of the key things there is that microbiome communities are like ecosystems like a rainforest or something like that, right? Where they can be in a state of balance, obviously. We all have them, and you can be an Olympic athlete and you have a microbiome, it's functioning well, everything is in balance. But it can obviously become dysbiotic. In other words, the organisms collectively move towards a state of imbalance where all the signaling and all of their relationships goes off. That can be really problematic. One of the biggest drivers of that dysbiosis is that a single pathogen gets into the atmosphere. So for example, let's say SARS-CoV-2 lodges in the lining of the gut and creates more of an inflammatory environment there. What we start to see is that the gut microbiome of those patients now starts to go off too, right? In other words, so the single pathogens can sometimes be like instigators that lower the immune or dysregulate the immune environment in a way that causes the rest of the microbiome to then encourages dysbiosis. So that's another thing that they do that's negative. The organisms in the microbiome on their own, it depends. I mean, they produce like a lot of neurotransmitters. They produce a lot of more of metabolites in the body are produced by microbes than even our own human genomes. I think in some cases, if we consider those relationships intelligently, we can even select for microbiome communities or certain ecosystems that produce metabolites in ways that are enhancing to human function. And in that case, I do think we can actually use the microbiome potentially as a tool to enhance longevity, but not really the single pathogens. Those, I actually think we just always want to mitigate their activity.
Could we even alter the microbiome in such a fashion to counteract the bad effects of the pathogenic microorganisms?
It's definitely a consideration. So for example, just one example is the vaginal microbiome in women with HIV. Women who already have a dysbiotic vaginal microbiome are much more likely to acquire HIV if they have a sexual contact with someone and they're exposed than if they have a balanced vaginal microbiome, and that's likely due to the fact that the vaginal microbiome secretes lactic acid and other compounds that can kind of, get in the way of other pathogens being able to more effectively colonize, right? So yes, it's actually possible that if a microbiome community is robust, that sort of prevents the entry or the ability of some damaging single pathogen to take over the community. So yes, it is true that microbiome health could mitigate the effects of single pathogens. Yep.
Thank you.
Wow. I think we have time for maybe one more. So I'll call on John.
Hi, I was wondering, in the case of persistent viruses that integrate into the human genome, such as the herpesviruses, whether anybody is looking into the possibility of using some sort of a genetic treatment like CRISPR to delete them from the human genome.
Yes, that is an area of research that's beginning. Some of the best teams are HIV right now, where they're doing a pretty good job of just trying to get HIV, which integrates into the genome. There's some good CRISPR-based approaches that are showing good promise. So yes, I think that CRISPR is going to be really important for pathogens for two reasons. To help with the reverse integration, and also in some cases to just directly break up the backbone. Because for example, when we see SARS-CoV-2 persist, the RNA is there in the tissue, and we say, what do we do to this patient? What we can do, we can give them Paxlovid with the drugs that we have. But the problem there is that Paxlovid prevents sort of entry into the cellular. It doesn't just get in the way of inhibiting RNA. And the thing is, a lot of these pathogens aren't spreading from cell to cell as much when they're latent or whatever. So we just need to be able to break up their backbones more. So basically, CRISPR-based approaches that can just directly get to the genetic material are huge. The biggest issue with those is that they have to be well targeted to the tissue type where the virus persists. So there's a lot of work on, okay, this has to get into the gut. How do we target this CRISPR? Or like antisense oligonucleotides are another one where you basically take the reverse of the sequence of what you're trying to break up or target, and you tag that, and then it hits the RNA and kind of mitigates it. That's another one we're looking at. It's getting them to the location where we want them. That's the biggest challenge right now. Or one of the biggest challenges. Yeah.
Thank you.
I think that's about it. Thank you so much, Amy, for your time and for the fascinating presentation. And do you have is there any way that like we can help you with your work, or is there any challenges or opportunities that you foresee in the next couple of years?
No, let's see. I think what I want to focus on, we want to put out some ideas for the first diagnostics that we think could be developed to help people track persistent pathogens and their impact on certain aging parameters as part of some of the health tracking that a lot of people are beginning to do. And I think that we could circle back to you guys with some of those diagnostic ideas, because I think that it's going to take diagnostics to make this real to people. We can talk about it a lot theoretically, but until someone can really measure that this is happening in them, I don't necessarily think there's going to be that much uptake in the longevity community. So that's what we're next focused on.
Sure. Okay.
So yeah, we'll be in touch. Thank
Please.
you.
Yeah. And feel free to share any papers within the thread of this seminar's thread in the email so we can follow up on that. Thank you, everyone. And we'll see you next month.